A baby born with a rare and dangerous genetic disease is thriving after receiving experimental tailor-made gene editing treatment. KJ Muldoon was diagnosed shortly after birth with severe CPS1 deficiency, estimated to affect 1 in a million babies. Affected infants lack an enzyme needed to remove ammonia from the body, meaning it can build up and become toxic. Researchers say KJ is among the first to be successfully treated with such a bespoke therapy and hope the technology can help many more in the future. And for more, let's bring in Fyodor Urnov, the scientific director at UC Berkeley's Innovative Genomics Institute and a person who knows this case extremely well. Welcome, Professor Urnov. You were involved in this whole process. There was no guarantee it would succeed. What was the breakthrough moment for you?
Oh, when Dr. Arns Nicholas said that the first injection went well. You realize until then, a giant team across 5 organizations, I don't know, maybe 200 people total, labored day and night for 6 months, racing against time. And to hear from Dr. Arns Nicholas that the first injection went well, and Kyle Jr. started to have protein again, I won't lie to you. I was driving at the time. I pulled over and I started to cry because all of us working on this gene editing CRISPR cure have been preparing for this very moment for a very long time. And the sense of exhilaration was frankly a bit emotionally overwhelming.
Oh, I can understand completely because KJ was, of course, facing a grim prognosis. Did you expect him to make medical history instead?
We were hopeful, but we were not certain. This could have failed in so many ways. You realize building a CRISPR gene editor is something any scientist in Germany or the States can do in their lab. But doing so to a quality where the US Food and Drug Administration allows you to inject it into a living baby is a completely different story. And the way we got this to work is first, we had sort of, if you will, a fellowship of a CRISPR cure. So we had multiple people and organizations who had specific superpowers. Dr. Arns Nicholas of CHOP knows how to care for children like that. Dr. Kiran Musunuru at Penn Medicine led the effort to make the CRISPR. A team here at the Innovative Genomics Institute, which was founded by Jennifer Doudna, she, of course, invented CRISPR gene editing, so it's a very moving moment for her, did the specificity assessment. And then we brought in leading industry companies. So Danaher, companies like Integrated DNA Technologies and Aldevron specialize in making the CRISPR to clinical grade. Then Jackson Labs made a special mouse which had KJ's mutation. Everybody was working in parallel. No one was sleeping. And honestly, the only thing we had in our minds was Dr. Arns Nicholas's guidance. If we wait until he's 8 or 9 months old, it's going to be too late. So when I heard from her just recently that he's sitting up in his crib and playing with his toys, I mean, what a ray of hope for all of us that people can come together to do something so incredible. But also, most importantly, Jennifer Doudna's vision for CRISPR is a genetic medicine for everyone who needs it. I think it's safe to say that for the first time since this technology was invented, we can seriously look parents in the eye who have children living with these diseases and say, we have entered a fundamentally new chapter in biomedicine.
This was an extraordinary group effort. You just explained it. But just for people who know less about the subject than you do, can this process be applied to other rare or even more common genetic mutations?
That is the beauty of what just happened. And this is why the sun is shining brighter. Jennifer Doudna's invention of CRISPR won the Nobel Prize, first of all, because it's scientifically beautiful, but also because there are very simple rules through which you take that magical thing, CRISPR, and you have it repair mutation 1 and then change it, very simple, to change mutation 2. And I can explain those rules to my 9-year-old daughter. In fact, I have explained those rules. Now, there's of course, a lot more to it. But we have never had a medicine where relatively simple principles can let you go from person 1 with mutation 1 to person 2 with mutation 2. So absolutely, yes, this is the beauty. I don't want to give families false hope. I've received 50 emails from parents since this story came out, and I've replied to every single one. I imagine Dr. Arns Nicholas received 5,000. The key idea here is we have now formed a new partnership between academia, industry and the Food and Drug Administration, where we now have a platform path. What does that mean? It means we can do certain things faster, cheaper, more scalably, because we've done it once. So we can point to KJ and say, see, this worked for him. It will work for the second baby.
Some much needed good news today. That was Fyodor Urnov of UC Berkeley. Thank you so much for your time, and congratulations to you and your team, and of course to KJ.
Absolutely. Thank you.
We're now joined by Julian Grünewald, who's an assistant professor of gene editing technology at the Technical University of Munich. So what exactly makes this case such a breakthrough?
Thanks so much for having me first. I think this is such a great day for the field of gene cell therapy and also for this special patient and the family, just because it shows that it is possible to actually develop a customized, a bespoke gene editing therapy for such a single patient with a unique mutation that is actually fixed with this new type of CRISPR 2.0 therapy.
So how far exactly are we from seeing this treatment available to other people? People in my own family have immunodeficiencies caused by genetic disorders. Could they be looking forward to looking for cures from this CRISPR 2.0 technology?
That's a great question. I think that's something that the whole field is striving to achieve. And I think what's so motivating about this for all of us is that it actually showcases for the first time that on a technical basis, it is actually doable. I mean, gene editing therapies are in clinical trials with hundreds of people, even for editing directly in the body. So this is very far evolved in terms of becoming an actual therapy. There's one CRISPR therapy on the market as well. But so far, all these commercial products have been therapies that actually help many people at once, which is a good thing. But what was so difficult to judge is, will it ever be possible to actually develop a therapy that's bespoke for a single person or a very rare mutation that only exists within one family? And what these scientists at UPenn, Kiran Muzunuru and Becca Arns Niklas, as well as Ben Kleinsteiber from Harvard have shown is that you can actually develop such a therapy in a very short period of time. And now to get to your question precisely, I mean, the major point is how can we find a way to have an infrastructure and also for a production and also a regulatory framework that allows to scale this to many more families and people. And this is a first proof of principle, but it's very encouraging. But this will, of course, take some more time. And it's absolutely crucial at the same time that we focus on how to do this safely for these individual patients as well. That's a paramount point about going about scaling this.
When it comes to gene editing, there are some detractors who say that we're opening up a can of worms when we start editing our own genome. So is this only a development that we should be looking forward to, or are there also ethical concerns that parents and physicians should be considering?
I mean, absolutely. The field is focused on looking at many different ethical aspects of this. I think what I would focus on now in this context and in this circumstance is that this is a therapy that's centered around so-called somatic gene editing. And that's something that's actually not debated in terms of ethical concerns. What is debated is editing the germline. And that's actually what I think most people in the field are relatively skeptical about and opposed to. And there's also a moratorium on this. But I think somatic editing, meaning editing cells in the body that will not be transferred to the next generation, to the offspring, to the children, basically, that is actually not that much of an ethical debate. I think what is really crucial for us to think about as society is how can we scale these therapies and make them available? Because for now, many of these very modern gene therapies are extremely expensive, and that makes it hard for us to scale them and get them into these families that need them. So I feel that actually these matters of accessibility and equity are also very important to consider in terms of ethical considerations.
All right, that was Julian Grünewald from the Technical University of Munich. Thank you very much for your information.